So, up until today, we've largely been talking about how genes work from an expression standpoint and the parts of genes, like exons and introns, and how they can be regulated. So, now we're going to talk a little bit about how all of that draws this connection between genotype and phenotype. Remember that the overall genotype of an individual is the sum of all of its genes. The overall phenotype of an individual are all of its observable physical and biochemical characteristics. Some genes have major effects on phenotype. For example, all of these genes have big effects, whether or not you have short versus long hair, or straight versus curly hair, or wire versus not wire hair, or whether or not you happen to have short legs or long legs. These are obvious phenotype differences that are caused by mutations in single genes. But the vast majority of mutations have very minor effects. For example, this is a SNP in one gene called HMGA2. On average, it causes you to be about half a centimeter to 1 centimeter taller than average. Having this single mutation, you probably won't notice that you're a little taller. Accumulations of many mutations that contribute to the same phenotype can result in dramatic changes in height, but individual mutations not so much. So let's talk about a specific genotype-phenotype connection. This is a classic example that's used a lot of the time in Mendel's experiments, where he talks about wrinkled versus smooth peas. The gene that's involved here, SBE1, encodes a starch branching enzyme. Essentially, what's happening to cause this phenotype is that the different chains of glucose are able to be connected in a side branch. When this enzyme is mutated, there's less starch. That means that these seeds absorb more water than they should. They'll swell initially and look larger when they're wet, but as they dry, they'll wrinkle. So the wrinkling is actually a side effect that's caused by this chemical change. The inability to create large starches with big branches creates this secondary effect when the seed is dry. A similar thing happens in wrinkled corn. For example, sweet corn. Sweet corn contains less fibers and more regular sugar, causing the corn to suck up more water as well. When sweet corn dries, the kernels also become wrinkled for the same reason. So, a lot of times, the phenotypes, the things that we see, have a molecular basis. The consequence of different types of mutations is what determines how the phenotype behaves. For example, most of the time, products or proteins will be made from both of the alleles for a gene. In a loss of function mutation, a null or amorphic mutation, we have a situation where the allele no longer functions. If two copies of the null allele exist, then no product for the gene is present. When one functional copy exists, half as much of the general product will be made. The question then becomes, what is the consequence of this? Having half as much. In a leaky or hypomorphic allele, instead of having no product, there's less product. In this case, there's a lot less. But in the case on this side, there's a little less. The question also then becomes, at what point does a phenotype appear? For example, if we were to think about biotinase efficiency, biotinase deficiency is failure to produce enough of the enzyme biotinase, which allows you to regenerate the use of biotin in your diet. This is a rare disease. For diagnosis, you have to have less than 35% of the normal amount. If you have more than 35%, you will have no phenotype. If you have less than 10%, you'll have the severe phenotype. This gives a situation where the leaky allele is likely to give either the phenotype or severe phenotype, depending on how leaky it is. The null homozygous will absolutely give you the severe phenotype. 50% as much would be considered normal. And depending on how leaky this is, it'd probably be normal too, probably close to around 70%. So what the outcome is will depend on when the phenotype would occur. We have special names for things like this as well. For example, if one copy of the functional allele is normal, we would call this haplosufficient. Haplo means half, and sufficient is enough. So half as much is enough. However, if 50% was not normal and instead conferred the phenotype, we would call this haplo-insufficient, where half is not enough. In a situation where haplosufficiency occurs, this looks like standard dominance. In cases where haploinsufficiency occurs, this often looks like incomplete dominance. So many times, how severe a phenotype is going to be based on how much of the product is removed by the specific mutation. And whether or not that mutation is recessive or dominant will depend on whether or not the threshold is met. Either there's enough of the product to be normal, or there isn't. In this situation, when we're thinking about it doesn't work, a null, this could be something like a full deletion, so deletes the whole gene. This could be something that is a single nucleotide change or a SNP, where we now have a nonsense early on. We could have a situation where we have a lost exon or retained intron that causes this. So now the protein is the wrong shape, so it can no longer perform its function, or we don't make anything at all. We could also have a situation here where the promoter is damaged, so we just don't get expression. There are lots of ways to result in nothing. When it comes to getting less, like these tend to be regulation mutations. They could be in an enhancer or a promoter. Sometimes they're in UTR regions. So the untranslated region is often used for recruiting materials to then translate these mRNAs. If that becomes less efficient, then we'll make less of the protein. It could be that it's in an exon, but it makes the protein less stable. So we make enough, but it doesn't stick around. There are lots of ways that you could get the same kind of outcome, right? So in this case, they're just talking about here's a mutation where protein is made at some point. Here's a mutation where the protein is never made. Depending on whether or not a little bit of protein is enough, we're going to get different phenotypes, right? So there are lots of ways to get the same outcome. What we're trying to get at here is that whether or not a little bit is enough is part of what determines the heterozygote relationship. So whether or not this looks normal or this has a phenotype. If it has a phenotype, if it's intermediate, which is often, then this is incomplete dominance. Gain of functions are often much worse. In a gain of function situation, a mutation causes the protein to now have a different function than it would otherwise have. Sometimes the original function is still there too. In this situation, we have a homozygous individual with the gain of function and a heterozygous individual with both a normal version and the gain of function. Other examples of gain of function can be expressing more than should be expressed for the gene. For example, we should produce 2, but instead we're producing 3. Again here, threshold will determine whether or not a phenotype occurs. Is 1 extra enough, or are 2 extra needed? Gain of function alleles can be good or bad. Gaining a function is not guaranteed to be helpful. For example, hypermorphic mutations, where there are too many voltage gated channels, can cause dramatic stimulus dependent behaviors like seizures. So, gaining additional expression of these types of channels is not good for you. So, whether or not a mutation is a null or a gain of function will also determine whether or not it could be dominant, recessive, incompletely dominant, or codominant, depending on the function of the protein itself. For example, let's say where plus is our wild type allele and M is our mutant allele. Normally, we're going to produce mRNA from each of our chromosomes, and both of them will be functional. In our heterozygote in this situation, we have one functional and one non-functional allele. If we get the wild type phenotype, then the functional allele is haplosufficient, meaning half as much function is okay, and the non-functional allele is not creating any problems. If having only one functional copy leads to the mutant phenotype, then this is haploinsufficient. One copy is not enough. And having two non-functional copies will confer a mutant phenotype as well. You should understand haploinsufficient and haplosufficient and how that can lead to what phenotype you see. In this situation, we would say that the mutation is recessive. In this situation, we would say that the mutation is dominant. So, what are some real world examples? PAH includes the phenylalanine hydroxylase, which is important for converting phenylalanine into tyrosine. It's one of the ways that we get tyrosine from our diet. Mutant alleles of this gene encode a non-functional enzyme. Without the enzyme, phenylalanine builds up in cells and can become toxic. This is what causes PKU. The normal PAH gene is haplosufficient. As long as you have one copy of the working gene, you're able to convert enough phenylalanine to prevent it from getting to toxic levels. So, this mutation would be considered recessive to the dominant working trait. It's haplosufficient. One copy is enough. For tumor suppressor genes, this is not usually the case. Most of the time, haploinsufficiency results in what's called an intermediate phenotype in the heterozygote. In the case of most mutations in things like repair genes, this creates some problems with being able to repair the DNA. It's not that you can't do it, you just don't have as much of the product that should protect you. This increases the likelihood that errors will occur. This is why carrying a BRCA gene, for example, can result in higher rates of cancer, even though you're heterozygous. You already have slightly higher risks of making errors, even though you can still repair.